So yesterday we talked about SDS PAGE, that is sodium dodecyl sulfate polyacrylamide gel, right? We will try to summarize this in 5 minutes or so that you can be updated, and if there is any questions you can ask me. So we know that this is your gel cassette, okay, where you cast your gel. Essentially is consist of your resolving, let me change the color code. Essentially it consists of your resolving or separating, and you have a stacking gels. There is a pH difference, right? 6.8, 8.8 or so. More or less they are same, but there is a pH difference. The percentage is roughly 4 to 5% of your stacking gel. The resolving gel is somewhere around your 8 to 10%. Okay, so you casted the gel, you put into the apparatus. Now there are different again apparatus comes, we will talk that in later on. So you put it into the chamber, you put your running buffer, and then you connect to your power source, and but don't switch on, but just you put in the buffer. Then you take a Hamilton syringe, or there are different pipettes which is there. You load your samples and then you run your gel. Roughly it is depending about again the speed and resolution and the buffer strength, like 160 volts or so is used, some like, or you can say 120 to 160 volts. Okay, and it might takes a time of 1 hour or so. And since you have a pre-stained marker, so you can also see your migration, and you also add in your loading dye the bromophenol blue, so you can also track how much it has run. This is an overall SDS page. Now it essentially consists of the separating or the resolving gel. It consists of water, acrylamide, bisacrylamide. Now again this differs to, but it usually made 30%. It is non-autoclavable. Store it 4 degrees Celsius for a month. Light protected. Tris hydrochloride means it is tris base, then you make up the pH. Okay, so usually tris base is used, and this stock solution SDS, because it's a SDS page. Then you add timid. Now this protocol is from Cold Spring Harbor, that's why they have used Biored from the manufacturers. This is the names. And then your ammonium persulfate, usually a 10% freshly prepared solution is made. Now in this about the stacking gel, as you can see, the 4% shown. Essentially same components, acrylamide, bisacrylamide, but then if you compare here, see, if you see the water, okay, and this is for a 10 ml typical gel which I am talking about. So this is more water, okay, because you are making it less percentage. Same portions is also added. First you will cast a resolving gel, then it will solidify, say, in half an hour or so roughly. Then you cast your stacking gel with the wells in this. Now the order in which you add, and the most important point here is the acrylamide and bisacrylamide. So acrylamide is as also written that it is a CN bonds, and the bisacrylamide is basically NCN bond, and it polymerizes. Okay, and this bisacrylamide acts as a cross-linking agent, and this process is you can say works in the presence of the APS, and the catalyst is the timid. That's why you add the timid at the later stage. Now depending upon how much you use this, then you can cast a different percentage of the gel. So this is how you see, this is a CN bond, okay, and this is a bisacrylamide, which is basically NCN bond, as you can see. Now so regarding your ammonium persulfate, so this is your ammonium persulfate, as you can see here in this picture, and this is your timid, which is tetramethyl ethylene diamine. So you can see there is 4 methyl groups, okay, these are the 4 methyl groups, and ethylene diamine which is present. Now if you look into the process in simple way, like when the timid comes in contacts with your APS, then it splits and releases the sulfide free radicals, and that helps in the process of the polymerization. Now this is a process which I then you basically run your gel, you load your samples, cathode, anode. Okay, now SDS is basically what is doing is it is providing the negative charge to the protein. So any proteins, all proteins, whether it is positively charged proteins like the basic amino acids containing proteins, or acidic is already there, or a neutral or proteins, they all will be charged negative. And the separation of your proteins over a gel is based on its mass. Okay, so the smaller molecules will actually move faster, and the larger molecules will be on the top, and you can see with a pre-stained known marker. I will talk about this in my gyromography, when we will be talking about the gyromography. Also we will talk about that how this process work, and there is a difference in the gyromography, that where you differentiate between because this is a SDS page, there you use a native page. Okay, or you can also use SDS page, but then you don't use what is called as staking gel, and for the reason I will tell in my next video. So this is regarding the mechanism, that how the movement occurs. Now the movement is based on the pK values of the glycine which is present in your running buffer. Now at a pH 6.8 or so, it's basically is a little bit negatively charged. Okay, so anything for any amino acid or any compound or any molecules, if it has a lower than pi value, it will be a positively charged, and but if it is higher than its pi value, then it will be negatively charged. I have a table to show you here also. Like if you see like can we use, because this was sometimes question asked, like can we use, like other than glycine, can other thing can be used? Yes, you can use, but the cost effectiveness and the molecule into consideration. Glycine is the simplest amino acids, and see this is the pK values for the glycine. So around 6.8 is little bit negatively charged, so that it can be moved. Now the process, as you can see in the diagram, the proteins which is in folded state, but when you add in a reducing, so there are two types of SDS page, you can say. The two type of SDS page is, one is the reducing, and this is number one, and the second number two, you can say the non-reducing. The only difference between these two is that you add the reducing agents like DTT or the beta mercaptoethanol into the sample loading buffer, but in this case you don't add it. And why that is so, I will tell in the next slide. It is basically you want to see certain subunits, so for that you use a non-reducing condition. So if you are using a reducing agent, say, which is usually followed, there will be disulfide bonds is break, your SDS basically change all the proteins into a negative charge, and then it migrates based on its size, which I just explained in a moment ago. So this is how its migration. So it is a staking gel, say a little more pore size, as you can see here. This is your separating gel, and the chloride ion which is at the top, then your protein molecules and the glycine. But as soon as it's reach your separating gel, then this glycines becomes number 2. If I say this is number 1, this is number 2, and this is number 3, then it will become number 2. This is number 2 1, this is glycine 2, and then 3, and then you get your proteins like high molecular weight on the top and then low molecular weight at the bottom. Okay, now this is a very if you are running a typical gel of 10%, then these are some of the proteins which is also used in your protein standards markers, as you can see. and you can see like if can as you can check your unknown protein or your known protein just to confirm everything is working fine or not. Like say for example, this unknown protein, based on its relative migration, you can find out what could be its molecular mass, right? So this is another picture to show you. This is based on reducing and the non-reducing, both are the same. See, this is the same proteins they are saying here, but when they are using a reducing agent, it means in terms of the sample buffer, and in another case they didn't, so there is a migration, you can say shift is there. This is another which I took from this paper. In this paper they have, if you just compare, we are not interested in like what these proteins are, but let's say just to show you the migration. So gel number 1, so this is your protein strain marker, shown the range. Say this is your albumin, okay, this albumin is lane number 2, but in the lane number 2 they are also showing that it is a reducing and a non-reducing condition. So there is not much difference, but if you see, compare the other parts, there are slight difference like in this particular cases. So that's how you can differentiate whether there is a subunits present. Like for a famous experiment, people use it for your insulin. So with this I will stop here, and